Hey everybody, welcome to the first video recorded lecture for Environmental Studies 282, Research Methods, Research Design in Environmental Science. These video lectures are planned to be serving the purpose of just being a good review for you from what we did in class, and or in some cases actually being the lecture content. Because we're so focused on building skills in class, especially skills with R and experimental design, we want to make sure our class time is available for you to work together and to work with me to actually try those new skills and develop them in class with feedback. So just a reminder, sometimes we're going to have these video lectures. I'm planning to record one of these for every single piece of content that we do in class. So you'll have a backup in the event that we do end up spending most or all of our class time working on R or working on experimental design. This class, lecture, I guess, day 2 of class is supposed to be focused on an intro to statistics and an intro to data and distributions. We're probably not going to get through both of those things. So I'm recording this video as your intro to statistics. So hopefully what you can see on your screen is what I can see on mine, which is just a very, very boring title slide about samples, variables, and intro to stats. So let's do it. First thing we're going to talk about is what is statistics? And if we were in class, which we're going to do this in class, I would make you talk to each other about this. So we do a little think, pair, share. You get in a group, you talk to each other. What statistics to you? Just come up with something that comes into your head. Like, what are the first few things you think of when you think of statistics? Hopefully you end up coming up with a nice list and you get to meet somebody and talk to them or talk to a group of people about what they think statistics are. Maybe you'll come up with something like this. From a textbook, statistics is defined as the study of methods to describe and measure aspects of nature from samples. A key component of statistics is that they provide us with the tools to quantify the uncertainty of measurements. Science is very imperfect. Nature doesn't really care about your hypothesis. So especially biosciences, we need to be able to quantify and understand our uncertainty. If I tell you that X is bigger than Y, it's probably helpful for me to say like the mean of X is bigger than the mean of Y. And there's some sort of error there, right? So when we're thinking about statistics, we're not thinking just of one number versus another number. We're often thinking of like a distribution of numbers versus another distribution of numbers. For example, if I told you that bluegrass was taller than green grass, I should probably tell you I sampled 1,000 bluegrass blades and 1,000 green grass blades that were all grown in the same conditions. And the average response was that green grass grew slower than bluegrass over a certain amount of time, right? So we need to be thinking about not just the comparison of means or the comparison of one number versus the other, but also like how does one whole population respond versus another? So that uncertainty is really important. And if that all sounds really confusing to you today, don't worry. We are going to spend our entire semester essentially understanding that concept. Before we dig into statistics generally, I just want you to have some definitions. So let's go over a few key terms. Estimation, the process of inferring an unknown quantity of a population using sample data. So if I'm given, say, a population of, I don't know, squirrels, and I want to know how big are their left front legs, I might look at, I might look at my squirrels and I could measure them, and that would be, a true measurement. But an estimation instead would be like I would look at a bunch of squirrels and say, okay, I've got like a large group and a small group, and I might guess like they're, one group is twice as big as the other, something like that. You've probably estimated a lot before. We all do. A parameter is a quantity describing a population. This is very, very vague. We're looking for some examples. Hopefully in class, you all come up with these. Something like the average, which we've talked about already. The variation of that average. Those are the kind of things that come to my mind. So average, variation, or average, standard deviation, standard error, something like that, the sample size. Any parameter that describes a population. We've been using that word population a lot already, so we should probably define it too. Population, it's all the individual units of interest, and then a sample is a subset of that population. So let's go back to what I was just saying earlier about the grass. If I wanted to know relative to one relative to the other, is bluegrass taller than green grass, or does bluegrass grow faster than green grass? I recognize that bluegrass is like a colloquial term for an actual species of grass, and green grass is just because I'm not very clever. So I just picked green. They're both actually green. That doesn't matter. In any case, a population of all of the grass in the entire world that was bluegrass is really, really hard to measure. Even if we're looking at like somebody's yard and they have like their front yard is one kind of grass and their backyard is another kind of grass, um, that's a whole population, right? The whole front yard, the whole backyard. And if they're each like an acre, which is a pretty large amount of space, I don't really want to go on my hands and knees and measure every single blade of grass. So instead, I will take a sample from that population because it's just a whole lot more reasonable for me, right? So if I'm taking a survey, here's another example. If we're thinking about, say, presidential election polls for the year 2024, what they'll do, most of these are still done over the phone. So that's obviously a bias, but they'll just start calling people and say, hey, who are you going to vote for? Or you can go in and you can fill out this form. Who are you going to vote for? They're not asking every single person in the entire United States or every single person in an entire state or county. They're just asking for anybody who fills it out or whoever they call to give them an answer. They're taking that sample and they're applying what they've learned from that sample to the whole population. And we're going to talk about how that works here in a minute. But population, all of the individuals in the unit of interest, all the individuals in some population of things, all the fish in Pasco Bay, all of the salt marshes in Maine, all of the, I don't know, littorine snails at Potts Point in Harpswell, something like that. So this is really a key component of statistics. It is our goal to understand the parameters of a population through something called representative sampling. We're going to talk about that coming up next. So we might think about what problems might arise from using samples instead of the entire population. The example that I gave of the presidential polling and how most of the big polls are still done via phone call, you might have a guess. They're going to be biased towards people who have time to answer their phone, people who like to answer their phone, and people who have, say, a phone, or even especially who have a landline. So lots of retired people are going to be answering these polls. And that isn't necessarily representative of the entire population, as you may have guessed. And zero millennials are going to be answering these polls because we hate the phone. I don't know about y'all, but maybe you also don't answer the phone. I don't know if Gen Z answers the phone. Maybe we can answer that question in class. But let's just assume that like younger folks don't answer the phone so much. Definitely aren't going to be part of these polls. So how can we limit these problems? Maybe we need to change the way we do these polls. Maybe they need to be a Google form or like, I don't know, a TikTok or like text me, something like that, right? If somebody were to text me like, what was my preference for a really easy yes or no question, I'd be able to give my answer. I wouldn't feel like super weird about it. So an example of how we might change the structure of our experiment to get better data. All right, let's dig into the idea of sampling and representative sampling. Sampling. We just talked about how in statistics, our goal is to take samples that are representative of our population. And here I am on the very next slide, tearing that down because this is imperfect. No sample is a perfect representative of a population unless it's the entire population. Sometimes this is due to random chance, which is what that's supposed to say, chance, but spell check. And sometimes it's due to sampling bias. So we might have sampling error, where the difference between an estimate and the population being estimated is caused by chance. Or we could have bias, a systemic discrepancy between the estimates we would obtain if the sample were truly the entire population, or if we sampled the same population again. So we're thinking like, does our sample look the same as the next sample and the next sample? And does it look the same as what the entire population would say? Are we going to know ever if we have this? We can infer a lot of things, but the answer is really no. Unless we are able to sample both the entire population and a sample and compare them, we're never really going to truly know. The problem there is like, obviously, we're taking the samples for a reason. We can't really usually estimate or get values from the entire population. We must estimate with samples. So we're probably going to have sampling error or biases. And it's not our job to never have them. It's our job to try to reduce them and to understand them and be honest about them as much as we possibly can. Statistics is a really useful toolkit, but it is not the be all end all. No statistical number ever is a guarantee of anything. So keep that in mind. So how do we reduce our sampling error and our biases? We think about our problem. We can't just say, I know statistics, therefore I know the answer, or like, I thought about this experiment, so it's perfect. Instead, we have to take our knowledge of the system, our knowledge of statistics, and our knowledge of our abilities, and also what biases may come up, what issues may arise when we do these experiments, and keep that all in our mind when we're doing our experiment. So essentially, we need to think critically and accept that we're not perfect. Here's our goal. We really, want to do a sample. Each one of these X's on our bullseye is an individual sample, where we take multiple samples and we compare them, and they're really highly accurate, which means they're close to the bullseye, which is what the true population would do. And they're highly precise, which means they don't vary a lot between each other. So when I said before, we're looking at understanding averages or mean responses or some value, and that value's error term, that's why we're looking at this. If all of our random samples have roughly the same average response and very low error relative to the other ones, then that tells us we have a really precise group of samples. And that gives us some idea that we're doing pretty well. And we don't know the whole population's estimate, but if all of our samples are pretty close, we might assume that we're doing a good job. Now, it is possible for us to go over here to the right, where we might have low accuracy right here, low accuracy and high precision. So that would say all of our samples are really close to one another, but they're not close to the true population. That's not super common, but it can happen, and we need to think about it. And then we can have these other examples as well, where we have low precision, which means our samples are pretty variable, but they're highly accurate, which means all of them are pretty close to the truth. They're just not very close to each other. And then, of course, we can have the old scatter shot, where we have low accuracy, low precision, and that just means we're doing a bad job. So, we're aiming for high accuracy and high precision, but, we don't always get there. So let's talk about some different ways of doing sampling so that we can think about how you might design an experiment. This class really is, at its heart, focused on experimental design. The first option is we can take a random sample. Random sampling is based on this concept. Each member of a population has an equal and independent chance of being selected. That means I go out in somebody's monoculture front yard and I'm trying to figure out how tall is the average, how tall are the blades of grass in the front yard? Every individual blade of grass has to have an equal and independent chance of being selected. So that means I'm not just picking the ones that are closest to me. I'm basically just randomly selecting any one of these. It can be really hard to truly randomize something. I'll get into that in a second. But random samples are allegedly there to minimize bias and make it possible to measure the amount of sampling error we have. So we can compare one random sample to the next, to the next, to the next, and we can get a sense for like how much different are our mean responses between our random samples. If we know that, we have some idea of our sampling error. Here's a couple of examples of random sampling, or simple random sampling in this case. You can see I have a population of people that are either happy, indifferent, or sad. If I randomly sample them, I should get something that represents the mean response of the population. And I've got 5 of each of my little heads here. So if I take 3 little people, I should on average get a happy, a medium, and a sad, or a happy, a meh, and a sad. And that is what happened. My sample is representative of the mean response of the population. So that's nice. Not as easy as it looks. The second example is more sciencey. Here on the right, what we have is a screen capture of a program called Coral Point Count. I'm going to move my captions. Coral Point Count, or CPCE, is a piece of software. You can load in an image, and it will, using a random number generator over the image, put down a bunch of random letters or dots. And each of those dots, the researcher is tasked with doing identification of what is under that dot. Using this can tell you who is in every image. So if you're taking an image every 10 meters underwater and every box is 10 meters, you can actually build data out of these images to tell you who is where. What is the percent cover, or how much abundance is there of these different organisms on this reef? So if you are swimming hundreds and hundreds of meters and you're doing this at tens of sites, you don't want to go in by hand and have to do all of this analysis. So instead, we use Coral Point Count to generate random points to help us build a model, a computer model that is then trained on our data and then can go through the data and do this all automatically, which is really handy. But this is an example of random sampling. It just uses a random number generator to place points at random. Another example of random sampling would be like I could take the names in the classroom and assign seats to you all, which I'm never ever going to do because I don't believe in that. But I could just put your names into a random number generator and have it place people in random seats. It's not super hard, but that's how random sampling might work. Okay, so how do we actually take a random sample? I just told you some examples, but say I don't know, we study river otters, and we're in a place where there's 75 river otters hanging out in a river, and they're swimming and playing and doing their thing. How do I take a random sample of those river otters? Do I just take whoever comes close? Is that truly random? Is there such a thing as truly random? Without random number generators, it can be really hard. So instead, we might do something kind of similar, but a little bit different. For example, we might try something like haphazard sampling, which is something that sounds absolutely ridiculous. And the more I explain, the more ridiculous it will sound. This is pretty much the standard in my field, is haphazard sampling. There's no systemic approach to sampling as haphazard. Here we are a couple of years ago, a couple of my students and I at Potts Point in Harpswell, and we're sampling with a transect the vertical extent from the high tide mark to the low tide mark of this rocky intertidal ecosystem. And we put out our line in one place that we randomly chose. And it's not random sampling, really, because we just kind of went. So this can be tricky. So this is an example of haphazard sampling. Another example might be you are in a forest and you're really interested in, I don't know, trees or leaf litter. Let's say we're trying to quantify leaf litter on the forest floor. And you have a transect, which is what you see Sam and Liam have a transect here, this little square. And they're quantifying everything inside that box. Transects, you use haphazard sampling a lot. The way that a transect will work, sorry, this is a transect here, this line. And then this little box here is a quadrat. So those of you who are already screaming internally, don't tell Holly that I messed that up. This is a quadrat. When you're doing quadrat sampling, a lot of times there's a system where you move along the transect tape, but other times you might use the haphazard sampling technique. And here's what you do. If you're out in the forest and you have your quadrat but no transect, and you're just trying to quantify leaf litter kind of at random in haphazard way, you will throw it over your head, and wherever it lands, like throw it backwards over your head, wherever it lands, that's where you quantify stuff. And then when you're done, you pick it up and you spin in 10 circles. You close your eyes the whole time, and then you throw it over your head again. Wherever it lands is where you quantify. That sounds utterly ridiculous and very non-scientific, but I can tell you from firsthand experience that this is very common. When I'm underwater doing surveys on coral reefs, the tried and true effective method for random haphazard sampling of a survey site is to put a point down, swim to that point, spin in 3 to 10 circles with your eyes closed, stick your transect tape down at that point, and then swim in the direction that you randomly spun. And then when you're done with your transect, you roll your tape up from that end point, you put your first point down, you spin in a circle 10 times, and you go in whatever direction you land. It is really just a way to randomly sample. And that you do that instead of building a grid. If you can't cover, let's say there's a grid of 20 or 30 transects, and you're just like, I can't do 20 transects in this site, but I can do 3. So you pick random ones instead of picking like a shape, unless, all within the bounds of the reef shape as well, right? So it's not really truly random. That's haphazard sampling. You may also try something a little bit more detailed, like this, stratified random sampling. That looks like what most people do in experiments. So population is divided into small groups called strata based on shared attributes or something. Like you might group, say you're studying like lobsters or something, and you have just a bunch of random lobsters. You might put them in size order, and then you might make bins of lobsters where you have like the small, medium, and the large lobsters, or you have like one of each small, medium, and large, or something like that. So you make these strata with members that have shared attributes, and then you randomly sample within that strata. So I take all of my large lobsters and I pick randomly from them, and then I pick all of my medium lobsters in a group and I pick randomly from them, and then I have all of my whatever's next and I pick randomly from them, and that helps me generate my random sample. This helps me reduce sampling error, and it gives me a chance to then like randomly choose individuals or groups from other groups. Seems like a useful way. It is relatively simple to do. And it's kind of already how our brains work. Next, we might try something like systemic sampling. And this is really common in survey work and in human subject work. You have a sample that's generated using a random starting point, but at a fixed periodic interval. Like you could say, oh, all of the people line up from, I don't know, line up in height order, or just line up on that line over there. So this is maybe how you pick teams, or like I coach Frisbee. Sometimes this is how we make our scrimmaging teams. We like line everybody up, and then we say, okay, we're counting. We're going to have 4 groups. Number 1 on the left, you're 1. Everybody count off, in groups of 4. So we'll go 1, 2, 3, 4. And then the next we'll repeat 1, 2, 3, 4. And that'll help us get a systemic sample of people in groups. So when might we use this in science? We use it a lot when we're working with people. What biases might it lead to? We kind of naturally group people based on attributes. So you have to be really careful about how this works if you're truly trying to be random anyway. And is it better than random sampling? For me, it's not, because we are almost entirely always, when we're making our first system, going to put people into groups, and then we're going to be selecting at a fixed interval from those groups, which means that we will have a really hard time keeping our implicit biases or our implicit assumptions out of our decision. So for me, systemic sampling, it's great when you're just trying to make groups, but if you don't really care that much about random independence, that is fine. Finally, we also have this other sample, a sample of convenience. This one, not so good. We really want to avoid these, but we're talking about them on purpose. It's a collection of individuals that are easily available to the researcher. You might be familiar with some of studies like this. Maybe you've heard of the Stanford Prison Experiment, or I don't know, anytime you survey a population of people that answer their phone, or a group of students in a class, or whatever. If you don't have a big enough sample size and you don't repeat the experiment enough times, a sample of convenience could be really, really biased, right? For example, if you look at presidential polling again, because it's the season, I've been looking at it a lot. If you look at different news outlets that have different slants, you'll see that basically their favored slant is always winning in their poll, no matter what reality is. Not that I know, but when you look at these different polls, you might see a really left-leaning thing saying like, oh, blue is up by 10%, and a really right-leaning thing saying red is up by 10%. The reality, according to the means, is like we're really neck and neck at the moment. So who knows? But given your sample of convenience, you're pulling from the people you know, from the people in your circles, that can obviously be biased. And again, if you're measuring all the grass and which grass grows the most, which grass is the fastest growing or has the highest height, and you pick only the samples that are closest to the path, they're going to be biased, of course, for some reason or another. Same if you go for trees in a forest and you're like, I'm only going to pick the trees that are close by, so I don't have to try that hard. Like, not trying is definitely going to lead you to have a biased sample. There's a bunch of examples on this slide of different convenience samples. So you've got your convenience sample here where you can see that the person in gray is just choosing the people that are nearest to them. And it's got the people colored in blue and green. So you can see that like, it just so happens that they're near a bunch of blue people. So as a result, the green perspective is almost entirely being left out. You may also do a kind of sampling called a snowball sample, where you talk to a person and you say, hey, can you like go find 3 people and talk to them and see what they say and report back? They're probably going to go out into their group and like find people who are like them for the most part, right? So you could lead yourself into these really biased sampling pools. This is basically multi-level marketing for sampling. A quota sample, where you say, okay, I know that there are 2 different perspectives. I have to take a green perspective and a blue perspective. So I'm going to talk to X number of green and X number of blue. Maybe okay, but you might also still end up with your sample of convenience where you only pick the people who are closest to you that fit in each group. It's still better than a straight up random convenience sample, but it's not great. And then purposive sample, you're seeking out, again, just like the quota sample, people from each group. But in this case, you're, instead of just picking the people nearest to you, supposed to be going out in different directions to find people, or sampling objects, species, whatever you're thinking of. None of these are great. You've probably done all of these, and that is okay. In class, we're going to try a sampling activity. We'd be sampling ourselves, and we'll choose some sampling schemes, trying to be accurate and precise. And we'll probably try to group ourselves in different ways just to see how it works. So hopefully we'll have time for that in class. I'm now going to go over some examples and just to summarize here what I mean when I say sampling. Again, after all of this, we've learned no sample is a perfect representation of a population. We choose sampling design based on a number of factors. Our goals are to be accurate and precise relative to the true population, and to get the best we can out of the resources that we've got. Nobody's ever going to do a perfect job because it just takes too much time, energy, and resources. We can only do what we can do. Our time is our time. But we are going to try to do the best we can with what we've got, which is just, a good way to live our life. In this course and in general in science, we attempt to use random sampling as often as we possibly can, but it's not always possible and it's not the only option. We have to keep all of this in mind when we're choosing how to sample, and we have to also recognize what biases we might get. All right, we're going to end today's video lecture talking about some examples. In biological sciences, and I know that this isn't necessarily only biological science, but that's my background, there's usually two different kinds of study. There's experimental studies where we do experimental manipulation, where we assign treatment groups, and we have explanatory variables to understand individual units. So the example that I'm showing you here in the top is a bucket mesocosm. So each one of those buckets is a small eelgrass meadow that we made. Each one of those buckets is an independent sample of eelgrass that contains some shellfish that we got from wherever. It's probably, in this case, they're mussels that are, they're oysters that are in there. So it's mud, eelgrass, and oysters. And they're replicates, but they're independent of each other. They're all subjected to different treatments. So the big blue barrels that you see to the top right, those are our treatments. One of them is hot water, so warmer than the average. One of them is the normal water coming in off the ocean. And the other one is water that has a high CO2 content, so it's acidified. We're testing the effects of warming, acidification, and control temperatures on the response of these eelgrasses and their oysters, how they grow, how they work together. In this case, we are experimentally manipulating just about everything. So experimental studies are highly manipulative versus observational studies, where in this case, Sam and Liam here are basically just saying, like, what's the difference in intertidal community diversity and structure across a whole bunch of intertidal ecosystems in the Gulf of Maine? So they went out, drove their vehicle out to all these different places in Maine, and they just threw down some transects and some quadrats and just quantified everything. They didn't manipulate anything. They just counted and observed what was there. So we have our experimental study, highly manipulative, and our observational study, where we basically just document stuff. So both are valuable. They're very different. And the way we sample can be very different. We'll talk about lots more examples of this as we go through the class. All right, here we go. Finally, ending up on just a couple of more terms. Types of data and variables. A variable, it's a characteristic that differs among individuals. Eye color, hair color, fingernail size, left big toe width, that kind of stuff. Categorical variables are also variables, and they are not numerical. They are qualitative characteristics of individuals that do not have a magnitude on a numerical scale. So we just talked about a variable. I gave you lots of examples. The ones that were categorical were eye color and hair color. In R, or in coding languages, we tend to call these factors. They're grouping variables. We also have numerical variables, which are quantitative measurements of stuff. So left big toe width, I said, would have been an example of a quantitative measurement. Seems straightforward enough, but important just to be on the same page. So in theory, I could give you this example. You're out in a forest observing trends. Name some categorical and numerical variables that you see in this image. Hopefully you can do that. I hope we have time for that in class. All right, most importantly, when we're thinking about data and variables, we're thinking about this. We need to build a hypothesis. Science is driven by questions. Hypotheses are basically those questions. We build a hypothesis around how one variable, called the explanatory variable, predicts or affects the other variable, the response variable. In a manipulative experiment, like the one we're seeing below, the treatment variable, the one being manipulated, is the explanatory variable, while a host of other variables might be the response variable. So here I told you we were looking at seagrass meadows. We're trying to understand survival over time. and maybe also growth over time. So those would be our response variables, and our explanatory variables would be the treatment, heat, control, ocean acidification. It's really, really important that we understand the concept of explanatory versus response. We're always going to be looking for that. So I told you the answer. I said, think about this experiment. What are some ideas? I already told you the answer because we're not in class. All right, when you're drawing a plot of response and explanatory variables, there is a, I'm going to say, field-specific colloquialism about who goes on what axis, what's on the X and what goes on the Y. I can draw you some examples, but generally speaking, when we're thinking about relative relationships between X and Y, we want our response variable, the one that is being measured to quantifiably change, to go on the Y-axis. And our explanatory variables will go on the X-axis or will be color or some such thing. Here's some real examples from 3 different real papers that I have published. Later on in the semester, you're going to learn that bar graphs are not appropriate for this specific use case, and you're going to learn why. But for now, let's just look. First thing we see on the left, I'm measuring the abundance of different corals at different sites that are, the different site types are classified by the color and also by the X-axis grouping. So you can see we have the green, the blue, and the red, and that they have different coral abundances. And those bars represent error versus the top of the bar graph itself. The top of the bar graph is the mean, and then the little T-bar, that's the error term. The rest of the bar is essentially meaningless, which shows you why bar graphs are bad. But nonetheless, you can still see the effect of X group on Y coral abundance. The middle graph here, we can see a couple of different variables. This is actually multiple dimensions. On the X-axis, we see the effect of year on extension rate. So how much growth does a coral get per year? You can see that in general that the linear regression lines in blue and red, or blue and black, are showing that there's a decline over time. All the gray is just squiggly noise from different annual bands. So we have 2 different panels here showing the effect of X on Y, and in 2 different sites, the nearshore and the forereef. So here you can see 3 dimensions of data in 2 dimensions. We'll get back into that when we talk about graphs later in the year. Further over on the right, we have an isotope biplot that shows how basically the relationship between carbon-13 and carbon-15 stable isotope concentrations. And here we're really just looking for relative difference, like how far apart are points from one another. This is a little bit of a different way to see data. And again, I'm just presenting this to you so you recognize that there's lots of different ways to do this. But the explanatory variables in this case are the colors and the symbols, and the response variables are both the X and the Y. So multivariate response. Okay, that's it for our first lecture on intro to stats and distributions. I'm going to talk about distributions in lots more detail in the next video and again in class. So hope to see you there, and I hope that this has been useful for you. All right, see y'all.